I'd like to thank my co-author, Wendy, who's breathed some new life into a long data set, and also Mary Power, who's helped all along the way, and many, many, many people who've come out in the field. Some of you are in the audience. I thank you. And if I've forgotten somebody's name, let me know. I became interested in this question about what happens when you subsample as a result of my involvement in FERC relicensing. And as you can see from this map, each of these little red triangles represents a relicensing site, and each triangle seems rather contained, but then what I've done is blown up what one triangle represents. It's not just one dam. Each relicensing involves many dams, many different reaches, and when you add them all up, it comes to a daunting number of river miles that need to be surveyed. And Ryan's talk was a great introduction for this. We've got a really big problem. How do we sample there, and what are the repercussions when we do subsample? And after all of these studies go on and a new license becomes effective, the data that are gathered during the licensing process become the de facto baseline data for assessing whether or not the new flow regime is having a helpful or a harmful impact on the frogs. So getting some certainty about how the estimates we make from these initial studies is very important. So subsampling seems like a necessity when the reaches are long and when access is limited by difficult terrain, uncooperative landowners, and we need to know what the consequences are in terms of our accuracy when we're making assessments of abundance, population trends, and the effects on flow regime. And so I'll be talking about those will be the three sections of the talk. Oh, I think I went too far. There we go. So to answer these questions, I'm going to be using a data set that we began collecting in 1992 at the South Fork Eel River on the Angelo Reserve, just a couple hours drive south of here. Each spring we search 5.2 kilometers of the river, and that's what's in dark blue. And we mark every Eggmass with a little popsicle stick and determine its fate. And as you can see, there's 5 different tributary creeks, perennial tributaries that flow into my study reach. And this graph here shows what the time series look like. On the y-axis, it's the total number of clutches found each year. And in 2005, we're almost exactly where we were when we started in 1992, and there's been fluctuations up and down in between. And I would argue that doing these Eggmass censuses is actually a good estimator of the effective population size because it tells us the number of reproductive females each year in the population. And I think it's really important to study a relatively stable population and say, this is the amount of hydrologic disturbance that a stable population seems to be able to recover from and what can be tolerated. Whoops, wrong way. What makes me a little bit queasy about subsampling is that I knew that there's this underlying spatial distribution of how the frogs utilize even my small reach of river. So what I've shown here is a schematic of how the breeding sites are distributed along the reach. And each of these bars represents a different breeding site, and the blue squiggly lines are where the tributaries come in. And as you can see, there's some clustering of the breeding sites near the tributaries. And near far, that's kind of a qualitative decision. But basically, about half of the population is breeding within one riffle pool sequence, either upstream or downstream of a tributary confluence, and the other half of the population is breeding dispersed in between the tributaries. And at the South Fork Eel, the pool spacing is about 5 to 7 bankful widths, and bankful width is about 35 meters, so that's a range of 175 to 245 meter radius away from the confluence, accounts for half of the population. Whoops, did I go too far? I did. Can we go back one? It doesn't seem to be working. Can we go back? Well, okay, we can't go back. I'll just tell you that when you do when you make a cumulative frequency plot, you'll see that that there's a small number of very populous sites. So in about 8 of the sites, it accounts for more than half of the population. But there's some problems with monitoring those larger sites. I really would like to go back. Okay, there we are. So there's some problems with monitoring the larger sites. We may miss the fluctuations because these larger congregations are less variable. What's plotted here is the coefficient of variation of the number of eggs that were laid at each of these sites over the 14 years. And on the x-axis is the mean proportion of the total population breeding at a particular site. And so what you can see is that the sites that are used infrequently, have small numbers of eggs laid there, have very high coefficients of variation. And so if we focused on just the most populous sites, we're choosing the sites that for whatever reason are more stable. It may be a function of the cross-section shape at those sites or just what's predicted by metapopulation dynamics, that smaller populations are more susceptible to stochastic events. But either way, I think that we can't just focus on the most populous sites. And even, and also finding those sites requires some full reach surveys. You have to have some sort of a priori knowledge. Okay, so what we've done is tried to estimate the accuracy of the abundance estimates you would make with 4 different kinds of sampling regimes. And our regimes are to base our searches on the tributaries, to have random locations, or have spatially dispersed random locations where you've got one random location sort of within each of the river kilometers, or take a mixed strategy where you would focus on, say, a couple of tributaries, randomly choose 2 tributaries, and then randomly choose 3 other sites. And so and then we tried to increase the search radius by increments of bank full width. So that's actually, just we've chosen that because it encompasses habitat units. As you go out 5 more increments of bank full widths, you've gone through another riffle pool sequence. And then we've estimated the total reach abundance from the number of clutches that would be found in the subreaches, and then we compared the predicted abundance to the observed. So this is what you get if you do tributary biased estimates. You way overestimate the abundance of the population. The yellow line is the actual mean abundance for the reach over the 14 years. And you can see as we increase the radius of our search area, it isn't until we've pretty much sampled the whole reach, starting at the tributaries, that we would get an accurate amount. So that you hit 10% accuracy only when the search radius has reached about, I think that's either 455 or 490 meters. And so if you think that's the radius, that means almost a kilometer for the diameter times 5 searches. So you're searching a lot of the reach. It's not really subsampling. Now, this is if you compare the random sampling, and you can see that the mean, this is the mean from 1,000 runs of a bootstrap simulation. The mean is pretty close right off the bat, although the standard deviations are large. And we could get into a debate about, at what point the standard deviations become acceptable, but you can see that you do a better job with random sampling. And then if you have random sampling that's dispersed, the means are very close, but the errors are less. And I think that's because essentially my tributaries are almost evenly spaced. So you're capturing more of the real system when you disperse the samples. Now, with the mixed strategy, I've just plotted here 3 of the possible combinations of, there's 10 combinations if you're taking 2 out of a group of 5, and I've just plotted 3 here. And your accuracy depends on which of the 2 tributaries you select. So you could either overestimate or underestimate abundance depending on which one you choose. But what if we're not really interested in knowing total abundance? Is there a way to select a subsample of sites to follow as an index population in order to detect trends through time? So instead of doing our simulation with abundance, we're doing it now on Lambda. And Lambda is defined as, it's the annual rate of population change. It's defined as the ratio of abundances in the same location measured at 2 times. So in our case, the location is the search area that's defined by our increasing radius. And so here are the results of how the tributary bias sampling does in terms of predicting predicting lambda. So the blue line is the lambda for each year that was observed over the study reach. In yellow, you actually get about a 10% accuracy when you're predicted from a radius of 105 meters. So that would be the equivalent of sampling 1/5 or 20% of the reach. You actually get a pretty good fit. As you increase the radius to 210 meters, so now you're sampling 40% of the reach, you can see that there's an even greater improvement in the fit. So now I'm going to get to the third section, where I'm going to talk about the goal of detecting the flow regime effects on population dynamics. So how and when would you expect the flow regime to influence population dynamics? Ryan and Hart have given a good background for that. And we could think that, so they've talked already about spring pulses, but I'm also going to talk about winter peak discharges. If there were effects of winter peaks on adults, we would expect to see that the next spring without a lag. The spring pulse effects would not necessarily become apparent in terms of population dynamics until these embryos grow to become reproductive females. So there's going to be some kind of a lag time. And there's also really significant flow effects on habitat, but they're going to have a much longer time scale. And if people want to talk about that afterwards or ask questions, I'd be happy to answer it, but I'm not going to go into it right now. So what's plotted here are is the hydrograph for part of the length of the time for the full year. And you can see that there's wet years with very high peak discharges. These are the mean dailies, and the little X's are actually the peak hourlies. So for the really big storm that we had in January 1st, 97, the peak's not on there. But essentially, we do not see that peak winter flows are not preceding any declines in breeding. The adults are effectively avoiding that kind of disturbance. But there is a significant effect of pulse flows on egg survival. This is the data from last spring, 2005. And each of the little green bars along the x-axis are the dates that I went out to sample. And then you can see how many clutches I found, and then I followed the fate of those clutches, found new, and the pie chart shows you the cumulative effect of those pulses. And there was 71% mortality. And it's not exactly straightforward, though, that you can say, oh, a bigger pulse causes more mortality. Clearly, the big pulse after Storm 2 caused the most mortality. But Storm 3, smaller than Storm 1, actually had more mortality because it happened later in the season. And there's a significant effect of, as the clutches age, they become more susceptible to scour. The jelly's less adhesive and cohesive. So here I have the spring hydrographs for my period of record. And you can divide the years into 2 types. Those in the top graph are ones where the maximum flow occurred when the eggs were laid. Each of these hydrographs starts at the date that eggs were first observed. So you'll see there's some variation in the date of when egg laying started. And there's also quite a wide range of the discharge when egg laying began. And this is in stark contrast to what Joe Drennan is going to be talking about later. And I really look forward to some interesting discussions about the differences between the results of natural selection and artificial selection for what discharge to begin breeding at. But either way, we've got two different types of years. The bottom years are years where there were pulse flows, maximum spring discharges happened after egg laying. And there have only been 3 years that were similar to last year. So it's a frequency of about 23%. So then when we look at how population growth rates are after those two types of years, there's no difference 1 year later. There's no difference 2 years later, but there is a difference 3 years later. And I would suggest that's the time that it takes for females to become reproductively mature. Even though males may reach reproductive maturity at age 2, the females have to be such a bigger body size. So that we can detect a signal from the spring pulses to the population dynamics. And despite that, I'll move on to my conclusions, and that is that the monitoring that's focused on tributary sites or relatively short subreaches would overestimate population size, but not necessarily population dynamics. In the absence of a priori knowledge about the longitudinal distribution that we would get from a full reach survey, it's really impossible to know if you would be accurately capturing the spatial and temporal dynamics of the populations. And our next step is to test the consequences of subsampling on our ability to detect the pulse flow effects on the population dynamics. And I'd really like to make a plug, or we would really like to make a plug for people with other data sets of longer distance, even if it's not a long-term, over many years. We'd really like to test this on a more heterogeneous reach where you do have a mix of slopes and a mix of different habitat types. Thank you.